The following is a presentation on the process of meiosis, which is the splitting of cells to create gametes, which are cells that can form a zygote. During interphase, the DNA in the cell is copied, resulting in two identical full sets of chromosomes. Outside the nucleus are two centrosomes, each containing a pair of centrioles whose structures are critical for the process of cell division. During interphase, microtubules extend from these centrosomes.
Prophase 1 occurs after interphase. During prophase 1, a multitude of movements and formations occur. This includes centrosome movement, spindle formation, and the condensation of chromosomes. In the early phases of prophase 1, each chromosome pairs with its homologue, and the homologous pair take part in something called crossing over, when DNA molecules of non-sister chromatids are broken and rejoined. These crossovers occur at a point called the chiasmata, where the chromatids cross over and exchange segments. While this is happening, the spindles continue to grow to form microtubules. Finally, right before metaphase, these spindles, or microtubules, will attach to the kinetochores of each homologous pair at the centromeres of each homologue. Metaphase 1 occurs after prophase 1. During metaphase 1, only a few events occur. First, the spindles and microtubules connect to the centromeres of each homologous pair of sister chromatids at the metaphase plate. Once attached, each chromosome in a pair faces the opposite pole. Both sister chromatids of one homologue attach to one pole, while the other chromatids attach to the opposite pole.
The next stage for the cell is called anaphase. In anaphase, there is a breakdown of proteins. These proteins are responsible for the sister chromatid cohesion among chromatid arms. The breakdown of proteins allows homologues to separate. The homologues then move toward opposite poles, guided by the spindle apparatus. The sister chromatid cohesion persists at the centromere, causing chromatids to move as a unit towards the same pole. After anaphase comes telophase. When telophase begins, each half of the cell has a complete haploid set of duplicated chromosomes. Each chromosome is composed of two sister chromatids. One or both chromatids include regions of non-sister chromatid DNA due to the crossing over which happened in prophase. While telophase is happening, cytokinesis is also happening simultaneously. Cytokinesis is the division of the cytoplasm. This is responsible for the formation of the two haploid daughter cells from the original one cell.
After the diploid cell splits into two diploid cells as a result of cytokinesis in meiosis I, meiosis II begins. Like meiosis I, the first process in meiosis II is prophase, but is denoted as prophase II. In prophase II, a second centrosome is formed in each cell, and spindles are formed from all four centrosomes. Towards the end of prophase II, the chromatids move towards the metaphase II plate. Evidently, prophase II differs from prophase I because the chromosomes no longer have duplicates, and crossing over does not occur. In metaphase II, the centrosomes move to opposite ends of both cells, and the chromosomes align between them. Then the microtubules from each of the centrosomes attach to the kinetochores of the three chromatids that face them. You can also see that metaphase II differs from metaphase I because the chromosomes that have aligned are not comprised of sister chromatids.
During anaphase II, proteins holding the sister chromatids together at the centromere break down, allowing the chromatids to separate. Spindles pull the chromatids to opposite sides of the cell. Afterwards, during telophase 2, nuclear envelopes form in each cell, and the chromosomes begin to decondense. During cytokinesis of meiosis II, each cell splits into 2 haploid daughter cells. As a result, the meiotic division of one parent cell produces 4 daughter cells. Each has a haploid set of chromosomes. In this case, because n equals 3, each daughter cell will have 3 chromosomes. Additionally, the 4 daughter cells are also genetically unique from one another and from the parent cell, because of the crossing over that occurred during prophase 1. It is by this process of meiosis that sexual reproduction is able to occur, and each parent passes down one allele per trait to its offspring. So how does the process of meiosis relate to Mendel's model of inheritance? Meiosis shows that Mendel's law of segregation holds true, validating the Mendelian claim that alleles for a heritable character segregate during gamete formation and end up in different gametes.
Thank you for watching our presentation on the process of meiosis.